The overall goal of this procedure is to produce sufficient DNA amplicon from a single microdissected polytene chromosome arm to use directly for fish experiments. This is accomplished by first preparing polytene chromosome squashes on specially designed membrane slides for microdissection. The second step of the procedure is to isolate and capture individual polytene chromosome arms by laser microdissection. The third step is to isolate DNA from the microdissected sample and amplify the DNA using 2 rounds of whole genome amplification. The final steps are to label the amplified DNA and to use them in fish experiments. Ultimately, results can yield copious fluorescently labeled DNA for a region of interest.
The main advantage of this technique over existing methods like fish with bat clones or PCR fragments is that large portions of chromosomes can be painted fluorescently with these labeled probes. Also, no sequence needs to be known for the chromosome paints, as they are mechanically isolated from intact chromosome arms.
First, isolate the polytene chromosomes, in this case from the ovaries of about 5 mosquitoes, fixed in fresh carnoise solution. Once a good chromosome spread is confirmed, the chromosomes are flattened by denaturation, followed by cold, overnight incubation. The next day, the chromosomes are dehydrated and washed to prepare them for microdissection. Be sure to clean the microdissection scope with ethanol and sterilize gloves and tubes with UV. Then, microdissect the chromosomes following a secondary video provided in the text protocol. Collect the DNA using a QIAGEN kit with a modification to accommodate an inverted tube. Follow this with column purification.